Good afternoon. I'm at the end of the session following Jane, Rob, and Andy presented the biorepository for brain tumors, which is located the same premises as NCARC and the Brain Bank. So for Royal, this is the red brick building down. The new building is not up yet. There's a hole in the ground, but it's going to come. We are part of the newly established Jeffrey Jefferson Brain Research Centre, which is a joint venture between the University of Manchester and Salford Royal to maximize the use of tissue and clinical annotation. The wealth of tissue and clinical annotation is available in the Neuroscience Institute of Salford Royal. We are under the general NCARC governance, but we do have separate ethics by University of Manchester for gliomas, particularly for investigating progression in gliomas, and then for pituitary, skull-base tumors and vestibular schwannomas. The second ethics also allow us collecting blood from normal individuals, from controls in tissue. We are integrated with the Manchester Brain Bank. The plan in the future will be of rotating staff in the biorepository and the Brain Bank to cover each other. There's also a plan, and we do have the ethics for collecting post-mortems for patients with brain cancer. Recently, we joined the Brain Tumor Northwest with Preston and Walton. Brain tumors are not particularly common, so we thought that working together will give access to a broader range of tissues and a broader range of clinical annotations. Dr. O'Leary, Claire, is developing a liquid biopsy arm of the biorepository as part of a UK consortium led by Susan Short in Leeds and part of an international consortium of GLASS. We do work very closely with Jane at MCRC Bank in a way that when a patient has an operation in Salford for a metastatic deposit, we do collect the tissue and then transfer Christie because they do have primary. They usually consent the patient when the primary tumor is operated on. We do not collect tissue from children, but we have links with CRG hospital and the Liverpool Alder Hay. So if scientists need tissue from pediatric brain tumors, we can put them in touch with the two biobanks. Briefly, what we do is identifying patients as suitable for research at the weekly neuro-oncology meeting in Salford. Then when we review the clinical history and the neuroimaging, then patients are consented either in clinic by the consultant neurosurgeon or the clinical nurses, or before surgery by the surgical registrars. As a neuropathologist, I'm notified when surgery happens, so we are available sort of on call and to maximize the amount of tissue that we can collect for research and minimize the time between surgery and the processing. Patients' data is anonymized. We process the tissue for diagnostic and parallel for research, tend to be present when the tissue comes down, so we are confident in selecting what is the best for the diagnostic, which always has the priority, of course, and then what goes in the biorepository. It's unlikely we collect biopsies because tissue is not enough for a final diagnosis and research. We tend to be collecting mainly from surgical debarking. Once the tissue is being quality checked, then it goes in liquid nitrogen and then snap frozen, and then it goes in a minus 80 for cryopreservation. As I said, we do process the tissue for diagnostic, but once the diagnosis is signed out and the patient is discussed following NDT, multidisciplinary meeting, then the tissue blocks become available. We tend to be fixing for 24, 48 hours, which is what is required for good DNA, but quality of RNA and protein is good enough for research, not as good as frozen tissue, but definitely good enough for PCR and protein analysis. Blood is collected in ADTA. We tend to be collecting a couple of mLs, and then we started collecting blood for RNA extraction as well. So we have 4 mL, 2 in EDTA and 2 in RNA tube. The EDTA tube, we spin the EDTA tube and then aliquot the plasma in 500 microliters and keep the pellet for genomic DNA extraction. Brief breakdown of what we have. It's not up to date. We have much more despite the COVID crisis. You will see the schwannomas are predominant because we are feeding the project, which is approved. So we do collect, in general, any patient who has surgery in Salford, but with priority to projects, to collecting for projects. As I said, we join up with Preston, Professor Dawson, and Dr. Prati Nitika at the Wharton Center. We are working toward establishing a unified ethics, where the patient has surgery in Preston first and then Wharton and then Manchester. We do not need pre-consent, and we don't need, we will not be needing a material structure agreement in place. It will be like a big biorepository at different sites. And a bit more challenging, we are working to establishing a common database with a common portal, not different from, very similar to what is in place for brain dementia research. Scientists can access the database, put a query into the system, see what is available in the 3 sites, and then apply for it. Briefly about the Preston, which is very more organized in Manchester. It was established about 14 years ago. This is a breakdown that relates to 2018, 2019, so they have more tissue now, but you will see they hold about 2,500 samples, including cells isolated from primary tumors, blood, serum, CSF. And as you see, the amount of requests is going up. As we do apply in Manchester, same as for Wharton Center, they have fees to charge for the processing for the technical work. But if scientists belong to the Brain Tumor Knowledge Consortium, tissue release is free. Briefly about the Wharton, again, there is a very good bank, quite organized. The person in charge is Kaja Sayed, who is a biobank manager. We are, our biobank manager resigned, so we are in the process of appointing one. At the moment, it's me, liaison with scientists. They do have uro-oncology samples similar to ours and Preston. They do also have tissue and blood from urology patients, particularly liquids, fluids, and they do have normal controls as well. Similar to our biorepository, they have quite a wealth of clinical annotations to complement tissue analysis. That's pretty much it. As I said, our biobank person resigned to go back to Australia. You will see my contact details in the leaflet that Alessandro and Becky circulated on Friday. Just feel free to contact me or email me should you need any tissue for brain tumor research. Thank you.